Hi, everybody. So my sister Kat has SLC 6A1, and as far as the science side of things, I am a research assistant at the University of Colorado medical campus, and I'm in a lab that's researching antisense oligonucleotide therapies, or ASOs. And so I'm going to explain a little bit about what those are. So before we get into what ASOs are, we need a little bit of background information just so that everyone's up at the same level of understanding. So as you may know, genes are used to make protein. And so that is done through a process called gene expression. And so with gene expression, you go from DNA to an intermediate molecule called mRNA, which is basically a copy of the same information, which that instructions are used to make the protein. So for SLC6A1, you would have the gene SLC6A1, and then the information would be transcribed down, which is then read by proteins to make a different protein. And so the process of how you go from DNA to mRNA is called transcription. So transcription is the level where ASOs would intervene. And so all you need to know about this big blue blob is that it is unzipping the DNA and essentially copying down the information into mRNA or RNA. And so before you get the final version of those instructions that you're going to use to make protein, you need to clip out all the unnecessary portions. So say when a reporter is taking down someone's story for the news, in their final version of the story, they're not going to include any of the ums, the uhs, or the buts. And so those pieces are cut out before you have your final set of instructions. And so if you see, we have the purple sections of the RNA as well as some gray sections there. And so the way those sections are cut out is through a process called splicing. So splicing is when you go from RNA to mRNA. So with normal splicing, you can see the important sections that you would like to keep for your final set of instructions are in the purple colored blocks. And then the straight gray lines in between them represent the pieces of information that aren't pertinent towards your final set of instructions. And so those pieces are cut out by cell machinery, and you have your final set of instructions there. But this can happen in different ways. So sometimes there's different forms of proteins where you include or exclude certain sections depending on what the protein is going to be used for. And this can also happen when there's a mutation that changes the splicing of a gene. And so a mutation could create a new place that's telling the cell to cut where it shouldn't, or that's telling the cell to not cut where it should. And so that is called alternative splicing. And so as you can see here, with alternative splicing, sometimes you would have that yellow bit and sometimes you would not. And say some mutations, you would always cut the bit out when you need it, or vice versa. And so this is where an ASO therapy would come into play. So ASOs would be able to interact with the mRNA either before it's spliced, during, or possibly even after. So what exactly is an ASO? An ASO is an antisense oligonucleotide, and that's a single-stranded piece of synthetic DNA or RNA that can bind to mRNA to increase or decrease the amount of protein made. It can also change the type of protein made. And now that's not an entirely different kind of protein, it's just a slightly different version of itself. So how do ASOs work? So first I'm going to explain some general mechanisms of how ASOs have worked, and ASOs that are known, the mechanisms are known. So here, an ASO can be used to degrade mRNA before it is used to make protein. So this would be useful in a case where a protein has a mutation that's causing it to be toxic to the cells. And you wouldn't want that protein to be made. So to avoid toxicity, you would use an ASO that would bind to it, and the cell would recognize that ASO bound to the mRNA and degrade it. You can also use an ASO to block certain proteins from binding to mRNA. So there are a number of different reasons why a protein would bind to mRNA. Sometimes it's to control when and where the protein is made, or even if it's going to be translated into, even if the mRNA is going to be translated into protein. So that can block cell machinery. Another way ASOs can work is interfering with splicing. So here I have a star to represent a mutation that is causing a piece of the RNA to be cut out that should not be cut out. And so an ASO would bind somewhere nearby or to a regulatory region, and that would cause a section to be included, and that would fix and repair splicing. And so the way that you control what the ASO does in the cell is by changing the chemistry of the ASO. So the ASO can bind to mRNA, but you would change how the ASO is formulated and produced to change what it is doing in the cell. So what to remember about ASOs in general is ASOs may not reverse disease progression. This really depends on what kind of ASO you're using and what kind of condition you're trying to treat. ASOs may reduce or prevent further disease progression, so it may slow things down and slowly repair function, but again, it really depends on a case-by-case basis. And ASOs are not permanent and will not alter the genome. And so even though they are technically a piece of genetic material, it is not interacting with your DNA. It is only interacting with your RNA. And since RNA is an intermediate set of instructions that is produced as needed and degraded as needed, it is not changing the master copy, the master set of instructions. So most of the ASOs that I've introduced so far depend specifically on what kind of mutation a person has. And we all know that mutations are not necessarily, they're usually unique to a person or a very small group of people. And so treating a large group of people with a single ASO is very difficult because they would all need to share something in common. And so developing an ASO for one person at a time is very costly, and it would be very time consuming. So to lead into how we would treat larger groups of people, we know that SLC6A1 is a haploinsufficiency. So in a normal cell, you would have two copies of your gene that are transcribed into mRNA, which is used to make protein. And so we have our two proteins there being produced from both genes. And then that's enough protein produced for enough normal function in the cell. However, when there's a mutation in one copy of the gene that gets transcribed into the mRNA, the protein that's produced from the mutated copy is either not being created or it's not providing enough function for the cell to be healthy. And so an ideal therapy would target the healthy side of the gene to increase the amount of protein made, whereas the ASOs that I had already presented are targeting the mutated copy of the gene to try to fix that. But if you could increase the healthy copy, you could potentially treat more people because it's not unique to the mutation. So how can ASOs be used to treat haploinsufficiencies like SLC6A1? The goal is to increase the amount of healthy protein to restore function. So you may be able to increase the amount of healthy protein in a cell. So this is something that we are currently researching at CU Anschutz. We're not looking into SLC6A1 quite yet because it's difficult to study because it's mostly neurological, and those kinds of cells are harder to keep alive and work with. So right now we're just trying to get a proof of concept in a platform. But the idea is that you would target a region that more people have in common with an ASO to hopefully increase the amount of protein that is made. Stoke Therapeutics also has something similar going on, and that's called TANGO. It's an acronym if you want to look it up. And so that's a similar idea of trying to increase the amount of healthy protein made. And so there's regions of RNA that contain instructions that say when and where a protein should be produced. And so those are regions that more people will have in common instead of an individual mutation that you're trying to fix. So I just want to repeat some main points. They do not alter the genome, and they leave the body like any other drug. And most ASOs treat one or a small group of people, and that is a problem that we are trying to fix. Because if you can treat the problem at the source and increase the amount of protein made, this will provide another opportunity to treat SLC6A1 instead of just its symptoms. And then again, researching ASOs takes time and resources. So it's really important to collaborate as a community and look into the science and just make sure everyone is sharing information so that we can help get this going as fast as possible. So before I end my talk, I just want to acknowledge my lab at my work, who helped me edit my presentation, and then SLC 6A1 Connect and Amber, who asked me to speak today, as well as my family, and especially my sister, Kat. So thank you.